to describe how to upload data using the Pangea web app. We're going to be following along in this guide in the documentation, how to upload sequencing reads to Pangea. This has all the same steps we're going to go through in this video, as well as a couple of other ways to upload reads to Pangea using either the command line or a more manual interface using the web app. So, to refresh, Pangea uses a data model that consists of samples. These could be a swab, a blood sample, something like that. These samples are grouped into sample groups. These sample groups and samples can both have what we call analysis results, and analysis results can represent the results of a bioinformatics analysis, like aligning reads to the human genome, or they can represent data like the reads themselves. So, to create a sample and to upload data to it, we're going to go to our profile. If you don't have a profile, you'll need to register, or if you have a profile but you don't see this button, you'll just need to log in. Once you're here, we're going to go ahead and hit this button to create a new organization. If you already have an organization that you want to use, you can just go to that instead. For this purpose, we're going to go through the whole process and create an organization. And all we need to do is input a name here. So we're going to call this MyDemo.org. Note, you can always change these names later. I also like to note that you have on the right here instructions for how to do this using the command line interface or in Python, if that's more what you would like to do. So we're going to go ahead and create that. This comes up. It has one person settings, but no samples, no sample groups yet. And I found it helps just refresh the page at this point. So this is no sample groups yet. We're going to go ahead and make a sample group. There are a few different buttons, a few different settings we need to add here. So first off, we need to give a name for a sample group. This should be your project name or something similar. So my sample group will do fine for now. We're going to want to give a brief one-sentence description. This should usually be a useful description. For now, I'm just going to say sample group for the demo. Some other settings here. Organization. This should already be set to your organization that you just created. In this case, it's MyDemo.org. You could select something else if you really want to. Public, we usually want things to be private off the bat, so we're going to leave this unchecked. But if you want something to be public, if you want to make data available to everybody, check that public button. Library, this means we can upload samples to the sample group. It's a little more technical, but typically you want to leave this checked. And use custom storage. We don't want to use custom storage, so we're just going to leave this unchecked as well. So once we've got all that in, really we just have to fill out the name and description. We're going to hit create, and we're going to get taken to this page with my sample group. And at this point, this is pretty much empty as well. So we're going to go ahead and create sample. So once you're on the create sample page, you're going to be on this page with this form, one sample with data. This is what we want to do. And there are a few different parts to this form. So I'm going to start off by explaining what those parts are. And then I'm going to actually show you how to use this and run a sample. So first we've got the name of the sample. This one's pretty self-explanatory, right? This should just be the name of your sample. It should probably match whatever you have in QBench, OpenSpecimen, whichever limb system you use. Then the data type for your sample, we have 3 data types available: paired end short reads, single-ended short reads, base called nanopore reads. If you have a different data type or a more unusual data type, you can still upload that to Pangea, but you're going to need to follow a little bit more manual process to do so, which is going to be described in that documentation guide I mentioned initially. So for now, we're just going to go with paired short reads. I just want to show everyone that if you collect single short reads, you get just one read instead. Pretty much the same thing for nanopore as well. So we're going to go share paired short reads though, however, so because that's what we're going to want. And we're going to see down here where we can select and upload the files. Now, the last piece of this form I want to go over is what we call work orders. Work orders are ways to tell Pangea that you want certain analyses run on your data that will then run automatically or mostly automatically off of your data and give you sort of a first view, first look at everything without you having to do anything at all. So those are the 3 parts of the form: name, data type, work orders, and then uploading the reads itself. So for a name, let's just call this my sample. Data type, we're going to leave that as paired end short reads. Let's go ahead and then select our files. I've got these very small Zymo control samples here, which I'd like to upload. They're just 1,000 reads a piece, so they're going to upload pretty quickly here. And then for work orders, I want to select the core quality control work order. This is, as you would expect, this is a quality control work order. This will run FastQC, some other basic stuff of it. And then we're going to go ahead and create an upload here. And that happened really fast. You can see that now we've created our sample. We've seen our raw paired short reads. We've got read 1 and read 2. If I click on those, they'll download, as you can see in the upper corner here. And then we're going to go back, and we're also going to see in settings that it's created a work order for core quality control. The status is pending because this job hasn't started yet. It should get picked up in just a second here. However, and it should go ahead and start. So now we can see that, yep, this is now running, and eventually this should turn green. It should run as well. Note, we can also add more work orders here. And actually, it looks like it might have already run. Fantastic, yeah. So now this quality control, this work order is run. We can see if we refresh the page that we've got another analysis result here with this FastQC results for this sample. So in this guide, we've seen how to do all of the steps of creating a sample, uploading data, adding work orders, and getting a little result out of this. Again, all the steps from this guide are here in the documentation for how to upload reads. And thank you all for watching.